Hey guys, so I'm sick, so I have to call it a sub today, but I want to go over the instructions for today's assignment. And also, I made a really big boo-boo. So I didn't notice I had made a mistake when I recorded myself during fourth period, and then I shared that video to everybody. So it was really bad. So let me go ahead and correct myself. Let me go ahead and show you guys how to do today's assignment correctly. So the first thing I want to do is I want to show you the example and correct myself on what went wrong. Okay. So today you're doing something very similar to the things that you did last time. Okay. So for what you are doing today is you are still doing protein synthesis. Our goal is to still make a protein, and we're still starting off with DNA. You're still going to have to transcribe into mRNA, and then we're still going to have to do translation to tRNA. But today's goal is we're trying to figure out if somebody has a certain type of sickness, okay? So it's still protein synthesis, but our goal is a little bit different. So before I continue, let me go ahead and show what I did incorrectly. So let's go over this first portion right here. And I actually highlighted the step that I did incorrect. So it says, use the DNA code to create your mRNA code. We're going to do that first. And let me actually change my color. Okay. So because we're going from DNA to RNA, oops, that's not a good color. Because we're going from DNA to RNA, we don't use T, we use U. Okay, so anytime you're going from RNA to RNA or DNA to RNA, as long as it involves RNA, we will always use the letter U. Okay, so first we're going to start off with DNA. When we are transcribing, we're going up and down. So from this one to this one right here. So T is going to pair up with A. A is going to pair up with U because we're going from DNA to RNA. C pairs up with G. G pairs up with C. C pairs up with G. And I'm going to continue completing this. And you will always go up and down. So whatever letter is here, you are pairing it straight down. So this would be G. A, because we're going from DNA to RNA, will pair up with U. G is going to pair up with C. And I'm going to go ahead and finish up this sequence right here. Okay, so that's the first step. We have completed step number 1. Now we need to go ahead and go to step number 2. Now, before we go on to step number 2, let me summarize why we just did this. So DNA has instructions on how to make a protein, but DNA is stuck in the nucleus. He can't leave the nucleus because he's too big, and also he's too important. He's doing a lot of important jobs. So we have our secretary, mRNA, who's going to go inside of the nucleus and is going to make a copy of the instructions on how to make a protein and take them to the ribosome. So that is the first step that we just did. mRNA just made a copy, and now she's on her way to the ribosome. So now that we're done with that step, we're going to go ahead and move on to step number 2. This one right here. So use mRNA to code for tRNA. And this is how it's going to work. We're still going up and down. So A is going to pair up with this one. So because we're going from RNA to RNA, we're still using the letter U. As long as we're involving RNA to RNA or RNA to, or DNA to RNA, we will always use the letter U. So A will pair up with U. U will pair up with A. We're still going up and down, guys. And we're going to finish this. The reason that it's in this little trio right here is because this is what we call a codon. A codon is made up of 3 bases. So now we're going to go on to the next codon. So C pairs up with G. G pairs up with C. C pairs up with G. So now we're done with this codon here. We're going to move on to the next codon. G pairs up with C, U pairs up with A, we're still going up and down, C pairs up with G. We're done with this codon here. Now we're going to go on to the next codon. U pairs up with A, C pairs up with G, G pairs up with C, and we're done with this codon here. We're going to move on to the last codon. A, because we're going from RNA to RNA, will pair up with U, U pairs up with A, and C pairs up with G. We are now done with step number 2. So let me explain why we just did that. So mRNA goes over to the ribosome and gives the ribosome the instructions and says, the instructions have all the information. It says, hey, you're making this type of protein, and this type of protein is made up of these amino acids. So he has all the instructions on how to actually make a protein. Now, the ribosome is going to use all of the instructions, but he's so busy building the protein that he doesn't have time to go get all the materials or all the amino acids needed to make that protein. So what he's going to do is he's going to call tRNA, and tRNA is literally going to bring him the amino acids, drop it off, and then go back and keep on bringing the supplies. That way, the ribosomes have all the supplies to build the proteins. The reason that we transcribe is because the ribosome is only asking tRNA to bring the supplies. He's not going to give him the whole list of instructions. He's literally just going to be like, hey, can you go bring me Val? Can you go bring me glycine? Can you go bring me histine? He's not reading him the entire instructions on how to build the protein. He's only telling him what he needs. So that's why we transcribe. So this is where I made the big boo-boo. And I don't know how I did it, guys. I guess I must have just been really tired that day. My apologies. But thank you. One of my kids caught it, and I want to make sure that I correct it. So it says, use the mRNA code on the coding. Use the mRNA code and the genetic code to determine your amino acids. So we're using our coding chart, but we're using mRNA, not tRNA. We're using mRNA. So what that means is we're using this section right here on the coding chart. And I'm actually going to highlight it so I can show you guys what I mean. So we are using these right here when we do our coding chart. So I'm going to go ahead and go to my coding chart, and I'm going to show you guys what I mean by that. So our first one is going to be AUG. So I'm going to go down to my codon chart, and I'm going to find A. So this is the first base. My first base is A. My second base is going to be U. And then my third base is G. So AUG is going to code for either MET or start. So I'm going to go back up, and I'm going to write start right here. I am now done with that amino acid. So now I'm going to go on to the next amino acid. So I'm done with this one, and this one. Anytime you use a codon chart, it will always use 3 bases. So here's my next one, CGC, and I'm still using mRNA, and I'm going to go back down, and I'm going to find C, G, C, ARG. So I'm going to go back up, and it's going to be A, R, G. Now I'm done with these. So now I'm going to go on to the next 3. G, U, C. So I'm going to go back down. G, U, C. It's going to code for Val. So I'm going to go back up, and I'm going to put in Val. Now I'm done with these 3 bases. So I'm going to go on to the next base. U, C, G. So U, C, G, SER. It's going to code for SER. So I'm going to go back here, SER, and I'm done with these. Now we're going on to the last one, AUC. And my apologies, guys. As you can tell, I'm sick. So AUC, going to go down here, AUCILE. So we've used our coding charts, and now we are done. So here's the steps that we did. DNA has instructions, so here's the instructions in DNA language. He has to get the instructions to the ribosomes. So he's going to call mRNA to come and make a copy. Here, mRNA made a copy of the instructions, and the instructions have what supplies we need to make a protein and also how to make the protein. So then mRNA leaves the nucleus, goes to the ribosomes, and tells the ribosome, hey, here's the instructions and here's the supplies that you need to make a protein right now. So the ribosomes are reading the instructions, they look at the supply list, and now that they know what supplies they need or what amino acids they need, they call tRNA and ask tRNA to bring them those amino acids. That's what this portion is right here. So tRNA goes, he's looking for the supplies, he brings the supplies, so that's this portion right here. We use mRNA to look at that codon chart, and brings the supplies to the ribosomes. Then the ribosomes can start building the protein. This is right here, this is a finished protein. And we're done. So that's the part that I did incorrectly. I accidentally used tRNA instead of mRNA. And I wanted to make sure that I correct that. So let me show you guys what your assignment is for today now. I'm gonna share my screen again. So what you're gonna do is you're gonna go to your class period, and on the very top where it says general, you're gonna go to assignments. And you're gonna see that you have one new assignment posted, and it's called 125 sickle cell anemia. So yes, today we're talking about sickle cell. And what sickle cell is a blood cell that isn't quite made right, and it causes a lot of complications. So before I go more into it, when you click on your assignment, you're going to notice that you can either do it on OneNote or you can do it on Word. You only have to do one. They're the same assignment. You can either do it on OneNote or you can do it on Word. Then you're going to see there's going to be a reference material. This is going to be your codon chart. So just in case, here's your codon chart. It's attached there. And this is going to be due on the 30th, which is this Saturday. So I'm going to go ahead and open up the worksheet, and I'm going to show you guys what it looks like. As you can tell, it's literally what I just showed you guys. So let me read and explain what sickle cell is. So sickle cell anemia is caused by a mutation in the gene that created the protein hemoglobin. Hemoglobin is the iron-rich protein that gives blood cells its red color and allows oxygen to carry from your lungs to your body and back. So let me make that into easier words. Every single one of your red blood cells has something called hemoglobin inside of it. And hemoglobin is actually the protein. Now, when somebody has sickle cell anemia, their hemoglobin isn't made quite right, and it can cause a lot of bad complications. In sickle cell anemia, the abnormal hemoglobin causes red blood cells to become crescent, moon-shaped, sticky, and stiff. So what happens is, if you think of your blood cells are nice and round. They're perfectly round. And this gives a lot of room to carry oxygen because there's a lot of room in that little circle to carry around the oxygen. Now, when you have sickle cell anemia, your protein hemoglobin is mutated. Something's wrong with it. So instead of being like a nice circle shape, it actually looks like a little moon shape. And let me show you guys what I mean by that. Actually, I don't know if I can. Let's see. So let me give you guys a good image of what that looks like. So this is a nice normal red blood cell. This is a sickle cell. So the mutation in the hemoglobin causes your red blood cells to kind of look like the little moon shapes. And the reason that this is so bad is because there's not a lot of room for oxygen. And because there's not a lot of room for oxygen, a lot of your body parts, like your toes and your fingers, they don't get enough oxygen delivered because there's not enough room to deliver the supplies. So we're going to go back to the Word document. So it says, both mother and father must pass the defective form of the gene for a child to become affected. If only one parent passes the sickle cell gene to the child, that child will have the sickle cell trait, but will not have any symptoms or negative effects caused by sickle cell. So what this means is both mom and dad both need to have that mutation in their genes. And then they can give it to the child, and that means that the child will have sickle cell. If, let's say, only mom has that mutation but dad doesn't, they'll give the gene to the child, but that gene isn't strong enough to affect it, okay? Because this is a recessive gene. So it's not strong enough to affect the entire body and make all the cells sickled. So sickle cell anemia causes life-threatening conditions such as an increased chance for strokes, organ damage, blindness, and much more. And the reason for that, guys, is because there's not enough oxygen going to the organs. There's not enough oxygen going to your eyes. There's not enough oxygen traveling, so it can cause a stroke. African Americans have about a 1 in 375 chance of having at least one gene associated with sickle cell anemia. It is routine to have infants tested for sickle cell anemia approximately 1 week after birth. So literally, like my child, even though I'm not African American, my child was still tested for sickle cell. Below you will find two infants, Alex and Nicole. Using protein synthesis, determine if they have sickle cell anemia. So this is how it's going to work. The very first one is going to be normal. So this is what it should look like. So you're going to make the proteins, and then you need to make sure that you're going to match the proteins. They have to be exactly the same protein. And then we have our two kiddos. We have Alex and we have Nicole. And your job is to tell me, does Alex have sickle cell anemia? Does Nicole have sickle cell anemia? Do they both have sickle cell, or does neither one have sickle cell? And you're going to do that by completing the protein synthesis. You know that one of them or both of them or none of them will have sickle cell if it doesn't match. So you're going to be comparing the normal to the kiddos. If you see that there is a certain protein or certain amino acid that doesn't match, that's an indicator that they do have sickle cell. This is your assignment for today. If you need the codon chart, remember it's under the reference material. Guys, I'm only an email away. I'm only a text on remind away. If you have any issues, please let me know. Have a great day, guys, and I'm sorry about my mistake.